Welcome to the e-learning content of Sri Sharda College for Women. My name is Gomati and I teach undergraduate students botany. This e-content is about DNA Data Bank of Japan, which is shortly called as Ddbj. Well, let's log into our class. Now, what you see here is exactly the home page of Ddbj. Now, what this e-content is all about is we are going to explore Ddbj. So in line with this, now let me try to share the screen where we can see the real content. Well, now what we see here is exactly the home page of Ddbj. Now, when you look at this page, you could see that this is about the bioinformation and Ddbj centers provides sharing and analysis service for data from life science researchers and advanced scientists. So when we scroll down a little on this page, we could see that there is a column called About Us because this homepage offers us so many things like search, services, activities, statistics, even super details on supercomputer. But before logging on to several things, now first we need to know what this DDBJ is all about. So therefore, I'm clicking here the About Us column over the home page. And now let us see what this is leading us to. Well, now what we are seeing here is the mission of DDBJ. So the mission says that this platform accepts research in biology today, which requires both computer and experimental equipment equally well. So here, the principal purposes of DDBJ is they say that this is a public domain where researchers can make their data open to the public, but through the INSD. Fine. And now here, this site records nucleotide sequences of living organisms, especially based on its evolution more directly than other biological material. So, scrolling down, we could see the governing structure of DDBJ and the major activities which includes construction and operation of INSTC, providing nucleotide and amino acid sequence data related to patent applications, management and operation of the National Institute of Genetics supercomputer system, and what are all the services that they are providing to search and to analyze biological data. Further, this site also offers us an important information like what are all the training courses and the publications that could be available through this DDBJ. But we need to note one point that the DDBJ Jing will exclusively be only in Japanese. Therefore, several sites of this platform will be in Japanese, hence has to have a good knowledge of Japanese knowledge language. However, we can also find a lot of information in English. So never mind, all your basic researchers or all your basic searches for researchers will be available through this training course platform. Well, now let us move on to the next page of this homepage. Now, what I'm doing here is I have come back to the same DDBJ homepage, and then you could see your option called search. So now here, this is the place where you can retrieve the data from the database. So now what I'm doing here is I'm clicking search. Well, you can also find a search button at the corner, at the right corner. Well, now here in the search button, now this gives us a lot of places where you can find ARSA, which is the annotated assembly data retrieval by using accession number and keyword search. And here, this DDBJ search includes the bioproject or biosample, SRA and JGA accession data by accession number and keyword. And here you can find taxonomy-based search. And here you can find that accession number database, which includes get entry. Fine. Now let me do one thing. here in the search column, I'm trying to type a nuclear, say for example, maybe let me type a nucleotide sequence for some protein, say for example, let me say albumin. Okay, and then now I'm giving a search button. Now let us see where this leads us to. See that no results. Search for albumin on Google. So now what does this mean? We will have to enter into every single site of the former column and then we will have to make the search. So now what I'm doing is I'm going to get back and I'm going to show you what is the right step to do. So well now you all can understand that how should a proper search should be. So now what I'm going to do is I'm going to enter into this Ddbj search. So now this Ddbj search is showing us a column like keyword, database like JGA, bioproject, biosample, SRA, and you can also find several types like biosamples, SRA samples, bioproject, etc. So now here is what I'm going to again type the keyword which we had mentioned it to be albumin. So now we could see that this DDBJ has found us more than 10,000 entries, which completes searching can be done in 31 minutes. So before going deep into it, now I'm going to click on this albumin. So this albumin has got this albumin is leading us to a next page where you could find the following. So here comes the page. You could see that this page provides you details like identifier, the type, and this comes under the bio project. What is the organism? What are all the properties? And then you could find several references. Here is a place where you can download the content, and they say that this status is available to the public and the visibility is unrestricted access. Therefore, they also give you details like when was this data created, when was this data modified, and when was this data published. Fine. So now what I'm going to do is I am going to enter into annotated assembly sequence. So before getting into it, now here you could see that this page offers you more information like you can analysis the vector screening system, you can analysis the web API for biology. This is the FTP site. Here you can find the annotated assembly sequence. Here you can find the sequence read archive, genomic expression archive, meta bank, bioproject, biosample, and this will also lead you to NIG supercomputers. Well, let us move on to supercomputer later. Now, let me show you how an annotated or assembled sequence will look like. So now what I'm going to do is I'm going to click this and I'm going to wait for the page to load. Fine. Now this has taken us to this page called DDBJ Annotated Assembly Sequences. Fine. So now what I'm going to do is, yeah, let me show you how to search using the keywords. So again, I'm going to submit to search using keywords. And now, I'm sorry. On to the same albumin page. And say, for example, let us look on to one Ddbj reference. So what I'm going to do is I'm going to click this bio project, and here you could find the title albumin, and you could find the description, and come on, look at the properties. You could find a lot of data with the programs in this page. Okay, and then if you want to download this content by clicking this HTTPS, you can download the whole content. So here comes the annotated sequence of the nucleotide what we have clicked. Fine. So now let me show you furthermore things on this database bank. Now let us move on further. So we were talking about the supercomputers. So this database also provides us details on the NIG supercomputers that is maintained by this DDBJ bank. So now here you can see the details of this NIG supercomputer, which is in action from 2019 till today, that is 2025. So here, if anyone has not seen any supercomputer, you can see the images. So this National Institute of Genetics, NIG, they provide a cutting-edge supercomputer system which is equipped with large-scale cluster-based computations, large-scale shared memory computations, and high-capacity, high-speed disk devices which could serve as a large-scale computational infrastructure for information processing in life science and medical research. Fine. So now here you could see the important announcement of this new system launch where all the informations are up to date, where even the power outage on April 7, 2025 um, has been informed. Now, there are, it is just a recent news. Just 3 days has passed for this. And you can also find a lot of instructions like changes in usage instructions and details on it. So here and how like here important notes on migration from previous supercomputers, etc. So here on the left side, you can find a lot of informations like information security policy, how the system architecture is made, hardwares, softwares, and database, and how to start using this NIG supercomputer system. So if anyone is interested, just merely by clicking onto this place where how to start using NIG supercomputer system, this will guide you to further details. So for this, they say that you need to register your account application before you start it. And even the details on how the account application can be made is given here. And like they also make you to take a pledge through email. And then after you get registered on this site, you will receive a certificate. And then you can log in into NIG supercomputers. So this is how things will be done here. Okay, fine. So now so far we have seen how the supercomputers could be used. I mean, how the NIV supercomputer could be used, and we've also seen that how the search could be done. Now I will take you to the same homepage where if you really wish to submit any annotated sequences or any data regarding your nucleotide details, now let me show you how things could be done. Now we have come back to the same homepage to the services center. So here you could find that these are the following services that this database is offering you. And here you can find, say for example, while going to biosample, here you can find the databases and submissions. So this biosample database contains description of biological source materials used in experimental assay. Now, if I need to do any submission, all I need to do is click on this biosample, and then here this gives you what the overview is, sample information, and what are all the validation rules. And here you can find sample attributes, say for example, and search and downloads. So now what I'm going to do is I'm going to click onto this overview of submission, which is taking me to another page where you can find details like what are all the samples, granularity, and then how the sample attributes, organism, the spatiotemporal annotations, and sample packages. Fine. See, here you can find that SARS-CoV-2 clinical or host associated. So these are the samples that are relevant to the public health. And here you can find that the wastewater surveillance samples that are related to public health. So this is how every single detail on this database can be accessed probably, and then like say for example, most of them could be accessed, but then here you can also find a few things which will peculiarly be only in Japanese. Fine. So now I hope you would have received an overall details on this homepage. Let me conclude this session by here you can find that this is the statistics of this database where how the database has grown. The database growth has made an exponential growth rate, and here you could see that by every year, from 2019, every year we could see that the rate of submission is increasing year by year. So this indicates the success of this database, which is holded by the Japanese website. And every year in all the aspects, we could see that there is an increase, which reveals that this database is offering great help to the public. I hope this video would have served useful to all of you. Thank you and meet you all later with another e-content.